they're using for aerobic respiration. At the same time, they're constantly making carbon dioxide waste, which is very toxic if the concentration gets too high. So they need a constant supply of oxygen, and they need to have their CO2 waste that they're creating constantly taken away. And of course, the circulatory system is what's doing that, at least at the cellular level. Air has about 21% oxygen in it. It's oxygen gas. So, and it's very low in CO2. It's about 1% CO2, although obviously that keeps going up and up and up as we keep adding massive amounts of CO2 to the Earth's atmosphere. But because we're relatively low in CO2 and relatively high in oxygen, diffusion is going to go the right way. It's going to allow oxygen to diffuse into blood and allow CO2 to diffuse out of blood. In water, the rules are different. Oxygen doesn't dissolve very well in water, and so it's never 21% oxygen, never even close to that. But the oxygen concentration in water can vary a lot. So that's a big problem, because the oxygen concentration is lower and it's variable. It's not really variable in air. Pretty much wherever you go on Earth, there's going to be right around 21% oxygen in the air. That's not necessarily true in water. Fick's law of diffusion is basically describing the rate of diffusion based on some parameters. And of course, if you think about it, we in a respiratory system, we want diffusion to happen as fast as possible to oxygenate the blood and get the CO2 out of it. So if you look at this equation, R in this equation is the rate of diffusion. And so natural selection is favoring R being a big number, being a large value. D is a diffusion constant, so this is not something that can easily be manipulated by natural selection. It depends on the material through which diffusion is occurring. So diffusion is actually faster in air than it is in water. A is the area across which diffusion takes place. And you notice that if A becomes a larger number, then R is also becoming a larger number because they're directly proportional to one another. So the more area you have for diffusion to occur across, the faster the overall rate of diffusion is going to be. Lowercase p is the concentration difference that you're talking about. In this case, it's between the inside of the organism and the outside. So if you have a high oxygen concentration outside the organism and a really low oxygen concentration inside, well, that means you have a high P. So the bigger P is, the bigger R is going to be also, because they are directly proportional. Lowercase d is the diffusion distance. It's the distance that a molecule that's diffusing has to travel. And remember, diffusion really only works quickly over really short distances. So as d gets larger, the rate of diffusion drops, and it drops dramatically. So you want d to be as small as possible to keep R as fast as possible, to keep the rate of diffusion high. So this is Fick's law. Now, the actual respiratory structures in organisms are being selected for based on the relationships that you're seeing in Fick's law. So evolution is optimizing the rate of diffusion, and it does that primarily by increasing the surface area over which diffusion occurs, so making A a large number, decreasing the distance that molecules have to diffuse across, so D needs to be a really short distance for R to be a large number, and by increasing the concentration difference. So you want a big concentration difference, because the bigger that difference, the faster the rate of diffusion. Now, if you remember, we already talked about surface to volume ratio, so you've already seen this slide before, but this is the problem that we keep running into. As organisms get larger, surface to volume ratio is going to drop, as long as the shape is staying the same. If an organism gets larger, the surface to volume ratio keeps getting smaller. And you need to have a large surface to volume ratio to allow diffusion to happen fast enough to keep the organism alive. So the way around the surface to volume ratio problem for large organisms is to not be made out of one big cell, but to be made out of zillions of tiny cells, and each of those tiny cells is, in some ways, it's working as part of a larger organism, but in other ways, each tiny cell is working as a separate tiny cell. Now, for this to work, we have to fill the spaces between these cells with a medium that has a high oxygen concentration and a low CO2 concentration. And of course, we're ignoring a bunch of other things that get transported in the blood, but we'll mainly just worry about the gas transport. And so remember, as an organism gets bigger, that surface to volume ratio drops really fast. So again, you've already seen this, but as the organism gets bigger, the surface to volume ratio dramatically drops. And so very quickly, the organism is going to run into problems if it's just getting larger, but nothing else is changing. So let's just take a quick look at the respiratory structures of a few different organisms. Single-celled organisms don't have any specialized respiratory structures. The respiratory structure is literally just the cell membrane. And that's because they're microscopic, so they've got a massive surface to volume ratio. They don't need to worry about adding extra surface area. When you get into larger organisms, you get into more problems. So you need to have extra surface area and to form a respiratory structure. That respiratory structure has to be really thin to keep the diffusion distance as small as possible. It really has to be moist to move oxygen into the blood and CO2 out of the blood. And obviously you need a large blood supply. So if you look at the organisms that are diagrammed, it kind of shows you where the respiratory surface is in the organism. And earthworm is kind of interesting. They've got they're not huge. They're huge compared to a single cell, but they're not huge as far as multicellular organisms go. But they have a relatively low metabolic rate, and so they just use their skin as their respiratory surface. So they've got a capillary bed beneath their skin, and that's where the gas exchange takes place. If you move over to the larval salamander, this is a fairly big animal. It's got a much higher metabolic rate than an earthworm, much lower than yours, but higher than an earthworm. So it needs extra surface area. Its body's surface area is just not going to be enough. And so it has gills outside the body that have to be incredibly thin, and of course they've got a big blood supply in there to allow for gas exchange. Insects have an interesting system. We've already talked about this a little bit. The respiratory surface for an insect is literally the cell membrane of all their cells. Their blood is not transporting oxygen, and so they are penetrated by this tracheal system, this system of air tubes. So here you're seeing the tracheal system going inside the insect, and so you just have diffusion between the literal gas that's inside that tracheal system and the cells themselves. Now, especially on larger insects and especially on insects that have a higher metabolic rate, diffusion through the air is not really fast enough. So insects that are flying are using a lot of oxygen, and for some insects are fairly large, so you wind up with a pretty good distance between the atmosphere outside the body and the length of the tracheal system that has to bring it in. And so you will see some insects where they're contracting their abdomen, contracting it. It looks like they're panting, and in a sense they are, because what they're doing is they're pulling their abdomen in and then letting it relax, and they are using muscles to force air in and out of that tracheal system to ventilate it faster than diffusion would otherwise allow it to happen. And then, of course, with the mammal here, mammals have really high metabolic rates, and mammals can be really large, and so they've got a really finely divided lung system inside the body with a lot of capillaries to move gases between the atmosphere and the blood. All right, well, let's talk about gills a little bit. Fish have a problem because, like I said, water doesn't contain a lot of oxygen at best. At worst, it can have really low oxygen levels, and so these fish have to be able to allow that limited oxygen to diffuse into their blood from the water. So they've got gills, which have a huge surface area. And so if you look at this, so you're looking at a bony fish. This is an actinopterygian fish here. This is a ray fin fish. So the water is entering through the mouth, and it's leaving through these modified pharyngeal slits, which are now modified into gills. And if you zoom in on the gills, again, you've got this theme of increasing surface area. First of all, each one is a little gill filament, so that gives you a lot of surface area because each one is kind of a little separate rod, each little bit there. If we zoom in with a microscope and look at each gill filament, you'll see that it's not a smooth cylinder. You've got little fins on them, which again massively increase the amount of surface area. All right, so here's the thing with fish gills. The water is always moving over those gills in the same direction, right? It's entering the mouth and then moving, across the gills towards the back of the fish. And so because there's always a direction to the flow, the water's always moving in the same way. You can maximize the rate of diffusion by basically maintaining a really high difference in concentration between the blood and the water outside the gills. And the way that that's done is with what's called a countercurrent exchanger. So we'll talk about that in just a second. Now, one thing that fish, you won't see any fish have is a lung, right? Because in principle, you can imagine, well, you could suck water into something like a lung, and just have the oxygen diffuse out and CO2 diffuse in, and then exhale that water and suck in some more water. Well, that doesn't work, and it doesn't work because the rate of diffusion through water is really slow compared to air, and because the oxygen concentration in water is really low compared to air. And so that kind of a system just isn't going to work. With a system like fish gills, you notice that you are completely ventilating those gills constantly. You actually can't ever clear all of the air out of your lungs, right? So you never clear out 100% of the air. You constantly have a little bit of stale air that's basically trapped in your lungs. It doesn't really matter because oxygen concentration is really high in air, and because the rate of diffusion is really fast in air, it's not really an issue. That is an issue for fish. So step one to increasing fish gill efficiency is have gills that they're not sacs. The water is just flowing past them, so they're constantly getting 100% fresh water flowing over them. Also, the gills are arranged in this countercurrent fashion. So take a look at the picture on the right first. This is not the way fish gills work. But here are those little fins on the gill filaments, and the dark blue ribbon here is the flow of water across the gills. And you've got the percentage of oxygen that's in the water. So obviously, whatever it is, we're calling it 100%. It's 100% of whatever it is. And of course, as the water flows over the gills, you notice that percentage goes down because, of course, that oxygen is diffusing into the blood of the fish through the membrane of the gill. Now look at the other arrow over here. This is showing you the direction that blood is flowing through this example. Now, like I say, there are no fish that have this, so this would be hypothetical. But notice that the blood inside the gill is moving in the same direction as the water. And if you look at this, you might think, especially at first, that, well, this is a pretty good system, right? Because we want to maximize the difference, the concentration difference outside the blood and inside the blood. Well, look at the difference. We've got 100% oxygen outside in the water and 0% in the blood. You can't get more than that. So that's great. But here's the problem. As time goes on, the oxygen concentration in the blood keeps going up. The oxygen concentration in the water keeps going down. When does diffusion stop completely? Well, it stops when those concentrations are the same. So when half of the oxygen that's in the water has diffused into the blood, that's it. Diffusion stops at that point. Or it doesn't really stop, but the rate of oxygen leaving the blood is the same as the rate of oxygen entering the blood, so you're not gaining anything at that point. Now take a look at the picture that's on the left. That's the way real fish work. So notice that the way the water is flowing over the gill is the same as in the first picture that we talked about. But notice that the blood is moving in the opposite direction of the water. That's why it's called a countercurrent exchanger, because the blood is moving in a counter to the way the water is moving over the gills. So when let's pretend we're following the water through here. So we start with 100% of the oxygen that's in the water. Well, if we look, we've got 90% of that in the gills. So there isn't a huge difference in the concentration, but we still have a favorable diffusion gradient, right? Diffusion is still going from high concentration to lower concentration. So it's the oxygen is still moving into the blood. As we're following the water, I should say, as it goes over the gills, notice that we keep having less and less, lower and lower oxygen concentrations in the water. Look here where it says 40%. Well, what's the oxygen concentration in the blood at that point? Well, 30%. So you notice that, again, it's not a huge difference in concentration, but we still have a favorable diffusion gradient, so diffusion is still happening. And when we get here to the end of the gill, when we've harvested as much oxygen as we're going to, we've got 15% of the oxygen left in the water. How much is in the blood? Well, 5%, right? We're close to zero, so we still are moving oxygen into the blood. And look at the difference after the water has moved over the gill. We only have 15% of the oxygen left in the water. Look at how much we had over here. 50% of what was there is still left in the water. So that countercurrent exchange system is really efficient at harvesting oxygen from the water. And of course, the same thing is happening, but in the opposite direction with CO2, is that's being basically allowed to diffuse out of the blood. Like I said, air has a lot of advantages to water when it comes to breathing. So the oxygen concentration is higher, the rate of diffusion is faster in air than it is in water, and so you don't need to ventilate the respiratory surfaces as completely in air as you do in water. So you notice with fish, you are ventilating those gills 100%, just constantly as fresh water is moving over them. That's not true for most animals that are breathing air. So we've already talked about this system, and I think you've already talked about it with Dr. Baker as well. Insects have a system of air tubes and air sacs, this tracheal system, that basically bring air directly up into contact with the cell membrane of every cell in the insect's body. So the cell membrane is the respiratory surface. And like I said, you will see this especially in larger insects, and especially like insects that have just been flying. They'll expand and contract their abdomens because that's squeezing on that tracheal system and basically ventilating it, forcing air in and out of it. So you're ventilating it better, faster than what would happen with just diffusion alone. Lungs are pockets with associated capillary beds that are basically inside the body. So they are essentially a whole bunch of surface area folded up inside the animal's body. Gills always seem kind of a risky prospect to me because gills are at least relatively exposed, and of course the surface of the gills has to be a really thin tissue for diffusion to happen very fast, and they're kind of exposed. But lungs, of course, you still have to have a very thin tissue, but at least they're relatively protected inside the body. So here you've got different kinds of lungs that some air-breathing animals have. So land snails have an internal mantle that acts as a lung. Spiders have those folded book lungs. Terrestrial vertebrates have either simple sacs like frogs. Frogs don't have a very high metabolic rate, first of all, and secondly, the lungs are not their only respiratory surface. Remember, they're also using their skin as a respiratory surface. So frog lungs are pretty simple. They're not hugely finely divided the way you might expect a lung to be. In mammals, lungs are highly divided, and birds have a very interesting system. Their lungs are actually not very large, but the way the air is routed through them, the air is always flowing in the same direction through the lungs. So think about it with your lungs. You cannot possibly have a countercurrent exchange system in your lungs because the air goes in and then the air turns around 180 degrees and goes back out. So you can't have a blood supply that's changing direction like that. But think about a bird lung. If the air is always going in one direction through the lungs, then you can have a countercurrent exchange system in a bird lung. Okay, so ventilation is using muscle power to move air in and out of the lungs so that you're not depending on that just diffusion, right? I mean, if you think about it, as long as your windpipe is open, your nose is open and exposed to the air, oxygen is diffusing down in there, but it's such a long distance and your metabolic rate is so high that it's just not doing you any good. You have to use muscles to force air in and out of your lungs to keep those respiratory surfaces well ventilated. Frogs have an interesting system for doing this. They have what's called a positive pressure system. What they do is push air down their windpipe into their lungs, and the way that they do this is by using muscles to lower the floor of their mouth, which pulls air in through their nostrils. Then they close their nostrils and their mouth, and then they use muscles to raise the floor of their mouth, which basically pressurizes the air. So now the air in the mouth is at higher pressure than the air in the lungs, and so the air flows down into the lungs. To release it, all they do is just basically open their nostrils, and the elasticity of the lungs, if they're just sitting there, will push air out, but they do have some muscles that they can use to squeeze the lungs a little bit with the muscles in their body wall also to force it out. So they are basically pushing air down from the top. Mammals do it very differently. Mammals have a unique structure, which, of course, because this is a human anatomy figure, they're not even bothering to show you here, but mammals have a muscular diaphragm right here, right below the lungs, and when it's relaxed, it's kind of in this curved shape. So what mammals have a negative pressure breathing system. So you are not, kind of swallowing air. You're not forcing it down into your lungs from your mouth. What you're doing is increasing the volume of your thoracic cavity, which, of course, if you increase the volume, and you've got a set amount of gas in there, the pressure drops because there's more volume now. And so now you've got lower pressure in your lungs than outside, so air flows into your lungs from the outside. But you're doing that by controlling the volume of the thoracic cavity. So you have a couple of ways that you can actually change that volume. So one way, which is what you do if you're just kind of sitting around usually, is that you contract these muscles of the diaphragm. Now remember, when it's relaxed, it's kind of dome-shaped, so if you contract those muscles, it pulls it straighter, and that increases the volume of the thoracic cavity. Now you also have some muscles on the ribs that will lift the rib cage, and so that's also expanding the volume of the thoracic cavity. And then when it comes to exhaling, again, if you're just sitting there, you're mostly just doing this with your diaphragm when you're not breathing really hard. And so when you relax the muscles of the diaphragm, it kind of goes back into this dome shape because of the elasticity. It goes back to that shape, and that reduces the volume of the thoracic cavity, and so you exhale. When you're actually, doing something, walking or moving around, and you're using more oxygen and your heart rate is up, you have muscles that won't lift all of your ribs together, but that going between the ribs that will pull the ribs closer together, so it pulls them kind of together and down. So you've got, again, two ways of squeezing down on the thoracic cavity to exhale air. All right, so down at the level of the alveoli, where diffusion is actually happening, you've got these tiny little balloons or little pockets. They kind of look like clusters of grapes, but they're hollow on the inside, and that's where the air is. On the body side of that membrane, you have capillaries, and so that's where oxygen is entering your bloodstream. It's in those capillaries. But like I said, you cannot have a countercurrent exchanger, because when you inhale, the air is moving into these alveoli and diffusion is happening. As you exhale, the air is moving out in the opposite direction. So there is no way that the blood can flow that's going to be the opposite of the airflow. It's always going to be, in the same direction sometimes and in the opposite direction other times. So this is not a terribly efficient system. Birds have an incredibly efficient lung system. And this is probably something that all of the dinosaurs had, not just birds. It's well adapted for flight because it is so efficient. And in fact, we know that, again, soft tissues don't fossilize very well, but we know from fossils that even the really big dinosaurs had hollow bones. They had air sacs in their bones, and that's what birds have also. So we think they had the same system, and it makes sense. So, if you think about it, a lot of these features that birds have that seem like it makes them really well adapted for flight, things like feathers and this incredibly efficient lung system and this efficient four-chambered heart, these have nothing to do with being a bird. These have to do with being a dinosaur. So they evolved long before birds did. So in the bird respiratory system, you have large air sacs. There is no gas exchange with the blood and the air through those air sacs. They are just like balloons. They're just storage areas for air. The lungs are small, and this is where the gas exchange takes place. And like I said, the air flows through those lungs in one direction only. It doesn't go through one way and then back up the other way. And it's actually not technically a counter-current exchange system. It's called a cross-current exchange system, but it works basically the same way. It's just that instead of the blood flowing completely the opposite direction as the air, the blood's flowing at like a 45-degree angle in the opposite direction to the air. So it's not quite the same, but it's the same. So this is massively more efficient than what you find in a mammal. Barred geese migrate over the Himalayas twice a year from Central Asia, and then they migrate down to the coast of the Indian Ocean in South Asia. So they are flying at around 30,000 feet elevation. And so think about that, because they're not just surviving at 30,000 feet with no extra oxygen. They are basically running Olympic marathons at 30,000 feet with no extra oxygen. So birds have this just incredibly efficient system. So here's kind of a diagram of it. This is very simplified. Birds have many air sacs. This is just showing a couple, but it makes it a little bit easier to imagine. If you were to look at the airflow through the nostrils, air goes in and air goes out, just like what's happening in your nostrils. So you might think, well, it's the same thing. But that's not true. So when the air is being pulled in through the nostrils, that air is not going to the lungs. It's actually going to the posterior air sacs and being stored there. That air then gets pushed through the lungs from back to front and goes into the anterior air sacs where it's stored temporarily. So as the bird, it doesn't matter whether the bird is inhaling or exhaling at its nostrils. In the lungs, the air is constantly flowing through. Fresh air is constantly flowing through in the same direction. When the bird is inhaling in its nostrils, what it's doing is filling up the posterior air sacs. When it's exhaling, it's emptying the anterior air sacs. But while it's doing that, some air from the posterior air sacs is moving through the lungs. So there's always air moving through the lungs. Now, like I say, what this allows is for this counter-current exchange system, which is extremely efficient. Even really ordinary birds, mallard ducks, which you see all over the place, they can easily fly for hours at 60 miles an hour. You just can't provide enough oxygen to keep your muscles going at that level of muscle use for any length of time, and it's just because our lungs are not efficient enough to do it. So this is a picture of bird lungs, or microscopic picture. So you can see it's just these kind of tubes that have a lining that's very spongy. And so the air is flowing through the tubes, and then you've got this massive blood supply around them that's picking up that oxygen very, very efficiently.